everyone, today we'll be covering the IGCSE Human Biology past paper. This is from 2021, May, and this is paper 1 as you can see here. So here the total mark for this paper is 90. So for paper 1 you'll be tested on the entire syllabus. And you are given, let's see how long it takes, you're given 1 hour and 45 minutes to complete this paper. Okay, let's start off with the first question. Alright, so it says the table lists 4 features of blood. It also shows 4 main components of blood. Put a tick in a box if the component shows the feature. Alright, let's start off with the first one, which is transport oxygen. So that would be your red blood cells. So your red blood cells contain proteins called hemoglobin, and oxygen would bind to hemoglobin and it would transport it throughout the body. Alright, the next part is has a nucleus. Now we know for the red blood cells that would be incorrect. It does not have a nucleus. And the reason why is because you want the red blood cell has been adapted to transport oxygen, which means they've basically what happens is that the nucleus is not there so that you have more space for hemoglobin. Now, the cell that does have a nucleus is the phagocyte. Let me just include a diagram of that. All right, so I've included a diagram here. So over here, we see the phagocytes, and you can see that they have a lobed nucleus. So that's very characteristic of a phagocyte. Let me just remove this. Okay. And platelets don't contain nucleus or nuclei because it's a very small cell component. I forgot to mention that the function of a phagocyte is to engulf pathogens like bacteria, for example. The next part is consists of 90% water. That would be your plasma. So the role of plasma is to transport all sorts of things like nutrient molecules like glucose, like amino acids, things like hormones, things like carbon dioxide. So because of that, it does contain 90% of water. The last one here is blood clotting. And that would actually be both the plasma and the platelet. So let me just include a diagram here. So this is the cascade that's involved in blood clotting. So these clotting factors are released by platelets. And then we have this thing called fibrinogen. And fibrinogen is a protein that is soluble in the plasma. So that is why both the plasma and platelets play a role in blood clotting. Let me just remove this. All right, let's go on to question 2. The diagram shows a model representing the arrangement of bones in the arm. Let me just zoom out a little bit. Okay, name the bones labeled 1, 2, 3, 4 on the diagram. So I'm going to draw an arm here, so bear with me. This is a thumb, so A is a thumb. And let me just draw it out here. And then we've got your 4 fingers, right? And then you've got your palm and your wrist. And then we have bones 2 and 3 is basically the forearm. And then this is like the elbow. And then you have the upper arm here at the top, which is what 1 is. So let's start from the bottom here. Number 4, this is your phalanges. Let me just write that down. All right, and then we have bones 2 and 3. So that's actually part of the forearm here. So 3 is the ulna and 2 is the radius. So your ulna is sort of near your pinky, which is this finger over here, whereas your radius is closer to your thumb. So that would be that. And then lastly, above your elbow, you would have your humerus. Let me just write that properly. All right, there we go. Okay, part 2, it says a hinge joint is a synovial joint. Draw a circle on the diagram to show where the hinge joint is found. All right, here, so it's referring to the elbow, and we just draw a circle to include all 3 bones. All right, part 2, name 2 other synovial joints found in the skeleton. So the term synovial joint is basically a joint, a movable joint. Let me just write that down. So we have a hinge joint, which is basically like our elbows, for example. It allows us to move our forearm up and down. So I'm just going to include a diagram here. There's actually several types of synovial joints, but you don't need to remember them. Although I guess in this case, it's a good idea to remember 3 types of synovial joints. In this case, we've spoke about the hinge joint already. That's at your elbow. You also have the ball and socket joint that we have here at the hips. So you can see that the bones in the legs are connected to the pelvis and allows movement in almost every direction. Here you have the pivot joint, so that's where our neck and head is at, and it allows for rotation. Okay, I'm just going to remove this. So I'm just going to write ball and socket, and then we have the pivot joint. All right, so in the marking scheme, it says gliding and sliding, or gliding/sliding. So let me just show you what that would look like. So here's the diagram. So in the marking scheme, it says a sliding joint or a gliding joint, and it is also known as a plane joint as well. An example of that is the joint that you see in the bones of our feet. Okay, let's go to part B. Vitamin D is important for the growth and development of healthy bones. Explain why vitamin D helps the growth of development of bones. So I've included a diagram here. This is from the textbook, but I don't think you need to know too much about this. But basically, vitamin D helps absorb calcium ions from our intestines to our bloodstream. And here we have a part of the bone called the compact bone. And you can see that these bone cells, or another fancy word for that is osteocytes, what they do is they secrete calcium ions in order for it to harden. So let's write that down. Vitamin D on cells in the bone growth. All right, let's move on to, let's go over the marks first. So one mark is for the absorption of calcium. The second mark is for bone growth. The third mark is mentioning the compact bone. All right, let's move on to question 3. Enzymes are protein molecules. Which subunits form enzymes? All right, so the answer for this would be amino acids because amino acids are the subunits of proteins. Part 2, it says describe how a solution can be tested to see if it contains proteins. So for this, we would use a Biuret solution. The original color is blue, and in the presence of protein, it would turn lilac. So let's write that down. So we'd say add Biuret solution to the solution to test if it remains blue, then no protein is present. If it turns lilac, then protein is present. All right, so the 3 marks are you need to add it to the solution. That's the first mark. If it remains blue, then no protein is present. If it's lilac, then there is protein present. Those are the 3 marks. All right, let's move on to part B. A scientist investigates the effect of temperature on two different enzymes, A and B. Enzyme A and enzyme B both digest proteins. It will digest it from proteins to amino acids. The results of the scientist's investigations are shown in the table. Okay, so temperature would be the x-axis and the highest is 90 degrees. So let's see if we can try and fit 90 degrees here. So we'd have, oh, there is no space for this, but make sure you try and draw within the gray box provided. So we'd write temperature, ooh, very small. And then we'd write 0 degrees, 10, 20, 30, 40, 50, 60, 70, 80, 90. Okay, make sure you include the units here as well in the axes label. All right, next we have the y-axis, which would be the rate of protein digestion in arbitrary units. The highest number that we have here is 230. Oh, sorry, it's 320. So let's try and see if we can fit that on the y-axis. So maybe let's go up in 50. So we've got 50 here. Let me just zoom in. 100. This would be 150. Do not give you enough space for this. 250, 300, and 350. All right, it's great. It's always important to start with 0. And we would label the y-axis rate of protein digestion in arbitrary units. I'm just going to write that down here. Oh, there's no space. Rate of protein digestion, arbitrary units. Okay, ideally you would have this positioned in the middle. Yep, so it looks a lot neater and within the gray border that they give you. All right, so we've got 0, 5, 5, 20. So 0, 5, 5, 20, and then 60, 80, 20. So 60, 80, 20, and then 0, 0. All right, and then let's, here it says draw the best fit curve for each enzyme. So I'm just going to draw curve. All right, and then we'll label that enzyme A. Well, actually, let's do the labeling later. And then next we have enzyme B. So that is 0, 10, 40. So 0 here, 10 here, 40 is here. And then we have 80, 140. Okay, so 80 is here. And then we have 140, which is over here. And then let's look. Okay, so 200 then 280. So 200 is here. And then lastly, 280. Then we have 320. So after 320, it goes all the way down to 80. So that's over here. Oh, over here. Okay. So I think it's a good idea to just label enzyme A first. Enzyme A. And then this one, ooh, this is going to be a doozy. and label this one enzyme B. All right, that's the graph. Let's move on to part 2. Describe the effects of enzyme B on increasing the temperature from 80 to 90 degrees. Okay, let's scroll back up to this graph so we can see for enzyme B at temperatures from 80 to 90 degrees, we can see that the rate of protein digestion has rapidly dropped. And that is because the enzyme is denatured at higher temperatures. So what that means is that the active site changes shape, and so the substrate no longer fits into the active site. Let me just remove this and write this out. All right, so enzyme B becomes denatured. When that happens, the active site changes shape and the substrate is unable to fit into the active site. So the 3 marks are denatured, that's 1 mark, and 1 mark for active site changes shape or loss of shape of the active site, and then substrate is unable to fit into the active site would be the 3rd mark. All right, let's move on to part 3. Describe the differences between the rate of protein digestion shown by enzyme A and enzyme B. Refer to the graph in your answer. So let's scroll back up to the graph. So we can see that through a larger temperature range, so from 20 degrees all the way to 90 degrees, enzyme B is a lot more active than enzyme A. So that's one thing you can also say. Another thing you can also say is that the optimum temperature is higher for enzyme B than it is for enzyme A. So for enzyme B, the optimum temperature is about 80 degrees, whereas enzyme A, the optimum temperature is near 60 degrees. Okay, so we can say from temperatures 20 degrees to 90 degrees, enzyme B is more active than enzyme A. Another difference is that the optimum temperature for enzyme B is higher at, at approximately 80 degrees. Whereas for enzyme A, at around 60 degrees. So the marks are B is more active. B is more active, that's 1 mark over a greater range of temperatures. So I wrote from temperatures 20 degrees to 90 degrees. And then the 3rd mark would be there's a higher optimum temperature for enzyme B. All right, let's continue on to question 4. The graph shows the percentage of people vaccinated against tuberculosis in a country between 1980 and 2010. It also shows the number of cases of tuberculosis in that country. So for your syllabus, you don't really need to know what tuberculosis is, but just to give you a bit of context, tuberculosis is an infection of the lungs. So it's a bacterial infection of the lungs. All right, so let's move on to the first question. So this is part A1. Tuberculosis is caused by the same type of organism as typhoid. What is that type of organism? Okay, so in your syllabus, you do need to know what causes typhoid. So typhoid is caused by a bacteria called Salmonella typhi, and that's a bacteria, so then tuberculosis would also be a bacteria as well. Okay, part 2. Describe the relationship between the percentage vaccination and the number of cases of TB. So TB is short for tuberculosis. So let's scroll back up here. So this part represents the tuberculosis vaccination, and this are the cases. So we can say that as the vaccination rate increases in the beginning, we can see that the tuberculosis cases decreases. We can even say that even though the vaccination rate is near 100% and remains constant at near 100%, the tuberculosis cases fluctuates throughout the years. So let's write that down. So we can say as the vaccination rate increases, the TB cases or tuberculosis cases decreases. If we scroll back up, another thing that we also notice is that even though the vaccination rates have reached about 100% in 1983, the cases for tuberculosis still continues to go down. So we can also mention that as well. As the are continue to go down. And we can write, although the vaccination rates stay constant at nearly 100%, the tuberculosis cases fluctuates throughout the years. All right, let's go through the marking scheme. So 1 mark is as the vaccination rate increases, the TB cases decreases. That's 1 mark. And then the second mark here, there is a lag in effect when it comes to this. So I mentioned that even though it's above 90%, cases still continue to go down. So there's a little bit of a lag there. So the third mark would be the third and the fourth mark would be these 2 points here. So even though the vaccination rate stays constant, the TB cases fluctuates. Yeah. All right, so let's move on to part B. So the population of this country in 2010 was 3.45 million. Calculate the number of cases of tuberculosis in this country in 2010. So we'd scroll up for this. And over here, 2010, we can see that this point here is 2.8, right? Because every point is 0.1. So 2.6, 2.7, 2.8, 2.9, 3.0. On the marking scheme, it says 2.75, but I don't think that point lies in the middle. I think it lies exactly at 2.8. So this is 2.8 per 100,000. So I'm just going to write that down. So it's 2.8 people per 100,000. And in this country, there is 3.45 million. So that'll be 3450000. So this is a ratio question. What you do to the right-hand side, you do to the left-hand side. So for this, we can divide it by 100,000 times by 3.45 million, and then we do the same on the other hand. We would also divide that by 100,000 and then times it by 3450000. So let me write that in the calculator. So 2.8 divided by 100,000 times by 3450000. And that gives us 96.6. So we can round that off to 97 people. So 97 cases. In the marking scheme, it says 95 people, but that's because they used a value of 2.75 per 100,000. All right, part C, explain how vaccination can give a person immunity to a disease. All right, so I've included a diagram here. This is from the BBC Bite-Size website. So what a vaccine has is this altered form of that pathogen. So that could be a weakened form of that pathogen, a harmless form, or perhaps part of the pathogen would be inside of the vaccine. So long as it contains the antigen, then it is able to stimulate an immune response. Now, what would happen is that you have your lymphocytes that would then produce antibodies, and these antibodies are complementary to the antigen that is present in the vaccine. And eventually what would happen is this would help the phagocytes engulf the pathogen. So the purpose of a vaccine, let me just bring another diagram in. So here's another diagram here. So the purpose of the vaccine is that if you're exposed to it the second time round, your body is able to produce antibodies at a much greater rate. So you can see the secondary immune response here. And the reason why it's able to produce antibodies at a much greater rate and at a higher concentration is due to memory cells. So let me just remove everything here. The antigens found in the vaccine will cause lymphocytes to produce antibodies that will remain in the blood. So then you can say when the person is then naturally infected by the pathogen, antibodies get produced a greater rate due to the presence of memory cells. All right, let's go through the marks. So one mark is vaccine contains a weakened pathogen. Second mark is that would cause antibody production. Third mark is and that will remain in the blood. Fourth mark is memory cells get formed, or the presence of memory cells. And then the fifth mark is antibodies can respond quicker, or antibodies are produced at a greater rate. I think that should be fine. All right, so for question 5, you can check out the next video. Let me know if you have any questions. Just drop them down in the comment section below. Bye!